Very good morning to all. Very good morning to all. This is Mr. C.H. Ajay Kumar, working as an associate professor at A.M. Reddy Memorial College of Pharmacy. And I'm dealing with the subject of quality assurance in the 6th semester, subject code BP6.60, which is very important subject for all the students, those who are going to complete the course of B Pharmacy, as they are going to entering into various manufacturing units. As they are going to enter into various manufacturing units in the different departments of research and development, and even in the teams of validation, the team of the calibration, and in the department of quality assurance and quality control. Students has to know the actual facts, what are the things happening in the pharmaceutical manufacturing units, and what are the various documentations that has to be done, and what are the various procedures that are going to be carried out in the pharmaceutical industries, as well as and they have to know the part of documentation and various quality control tests and various in-process quality control tests. As a part of this, we are going to study about the unit 5 in the quality assurance, that is validation and calibration and various principles of validation and validation master plan, and what's the difference between the validation and the qualification. And in the part 1, we have discussed about the validation master plan and the importance of the validation, the scope of the validation. And today's session, we are going to discuss about the calibration and the qualification of various analytical instruments by taking a few examples. And also we are going to know about the warehouse. Let us start with this topic. So we are going to know about the calibration of pH meter. So the pH meter is used to measure the acidity and alkalinity of a solution, or a pH is an electronic device used to measure the pH of any solution. So in order to know what is the actual meaning of the calibration, here many of the students use it to give the answer that the calibration is nothing but checking the instrument whether it is working properly. But according to the actual definitions given by various bodies and various like ICH, USFDA, calibration is a process of checking the instrument whether it is giving the results in a predetermined manner, which are very accurate, and whether all the adjustments has been done and giving the predetermined results. And without calibration in the pharmaceutical industries, no activity is being performed. And even we can't we can't even use any kind of equipment for the process of measuring of different values such as moisture content, pH, or the weight, or specific optical rotation, or moisture content. Any other values which are very important in the pharmaceutical industries, by using various equipments or instruments, has to be done only after performing the calibration based upon the calibration frequencies. So see here, the calibration of the pH meter that we are going to discuss today. From the 5th standard onwards, we are listening the definition of the pH. What is meant by pH? The negative logarithm of H plus ions concentration is nothing but the pH. So here the pH meter works basically by knowing the changes in the H plus ions concentration in the given sample. What we are going to calibrate here in the pH meter? We are going to calibrate the function of the electrode that are being used in the pH meter and the functions of the pH meter equipment. So here the pH meter is used to measure the acidity as well as the alkalinity of the solution. Or the pH meter is an electronic device which is used for measuring the pH of any solution. So the pH meter calibration is required based upon the frequency, that is 24 hours frequency. For every 24 hours before taking the first reading, the pH meter has to be calibrated, and as per the defined frequency, that means 24 hours frequency, and it has to be maintained in its accuracy. So here the calibration of a pH meter is a 2-point calibration method which is performed using two buffers of known pH. What are the two buffers that we usually prefer? Why we are calling it as a 2-point calibration? That means just we are simply going to measure, or we are going to calibrate with the two different standard buffer solutions. So thereby it is being called as a 2-point calibration. One of the standard pH buffer is pH 7.0 and other is either an acidic or alkaline buffer of a known pH. From where we are going to purchase these buffer solutions, standard buffer solutions, and how the calibration has to be performed for all this by using all these standard buffers. Usually in the manufacturing units, utmost care will be taken for the process of calibration of pH meter. And the stability of the various pharmaceutical products lies upon the pH of maintained in that particular pharmaceutical product. So one of them is a pH 7.0 we are going to take, and from the NIST, National Institute of Standard Technology, they are preparing the standard buffers which are much more accurate. So for example, if I am entering into the pharmaceutical industry by 9 a.m., when I am rushing into the QC laboratory in my manufacturing unit, if I want to check a sample which has been received by CRD department or any other validation team, obviously I want to use the pH meter for the purpose of measuring the pH of the sample received from the chemical research and development or any other validation or any other departments. Obviously, I will look into the logbook of the pH meter. In that logbook, we have to see whether it is being calibrated or not. If the logbook is mentioning that no calibration has been done, I have to do the calibration. And I have to give the status of that particular instrument that it has to be calibrated. After calibration, I have to give the tag that it has been calibrated at particular time by using various standard buffer solutions by mentioning the batch number of the sachet of the standard buffers which I have used for the purpose of calibration, and at what time I have started the calibration, and what is the standard buffer value, and what are the results that have achieved after performing the calibration. Okay, so these all the details has to be included in the logbook of the calibration of the pH meter by mentioning the equipment name and what is the make, what is the model, and what is the ID of the equipment or ID of the pH meter. So it is important to make sure that all the buffers at the same temperature before beginning the calibration, because pH often varies with the temperature. So coming to the process of calibration, Switch on the pH meter and wait for enough time to get warm up. Remove the electrode from its storage solution. Rinse with distilled water thoroughly and blot dry using a piece of tissue paper. There is no need of rubbing the electrode tip or electrode bulb. Rubbing makes decreasing the sensitivity of the electrode. So as all the electrodes that we use for the pH meter are very sensitive, and they are very sensitive glass membrane electrodes, as because the glass electrodes that we use are having the property of both reference as well as standard qualities. And place the electrode tip, which we call it as a bulb, in which the bulb is said to be sensitive towards the changes in the pH or the changes in the H plus ions concentrations or OH minus ions concentrations. Place the electrode tip into the buffer solution of the pH 7 and press the measure or calibrate button and wait until for the display to get stabilized. Adjust the calibration button to make the display reads 7 if required. Remove the electrode from the buffer solution, rinse with distilled water and blot dry using fresh tissue paper. Place the electrode into buffer solution of either pH 4.01 or pH 9.20. One is the acidic buffer and one is the basic buffer, and wait for the display to get stabilized. And adjust the calibration button to make the display reading as pH as 4.01 or 9.20, depending on which the buffer that you are using or you have used. Remove the electrode from the buffer, clean with the distilled water and blot dry with the tissue paper. Place the dried electrode back into the storage conditions. So when we are measuring the pH for the buffer solution of the 7, it has to give the results of plus or minus 0.02. Okay, that means, for example, if we are measuring the pH of standard buffer solution of the 7, we have to obtain the results of 6.98 or 7.02. If we are measuring the pH of 4.01, we have to obtain the value of 4.03 or 3.98. Or if we are measuring the pH of 9.20 standard buffer solution, we have to obtain the value of 9.22 or 9.18 plus or minus 0.02. If the values exceeds these limits, then the calibration is said to be failed. If the values are within the limits, then the calibration is said to be passed and it is ready for the measuring of the sample solutions. So, and we have to give and tag that, and we have to mention in the logbook that the instrument is said to be calibrated at particular time, and we have to give an authorizer signature by whom the calibration of the pH meter has been performed. Okay, so this is all about the calibration of a pH meter. And coming to the qualification of heavy visible spectrophotometer. In the earlier classes in the part 1 of the unit 5, when we are discussing about the validation and the qualification, there is a narrow, very narrow difference between the qualification and the validation. Validation is especially for the procedures, methods, and the systems, utilities, and the facilities. But whereas coming to the qualification, which is a part of the validation, qualification is especially for the instruments and the equipments. So before discussing this qualification, we will come with a discussion of qualifying any equipment, qualifying any equipment of our routine use. For example, if we are purchasing an any electronic gadget such as the mobile phone, we used to perform this qualification. If we are purchasing any home appliances such as the refrigerator or air conditioner or any geyser, we'll perform the qualification. If you are purchasing any, like laptops, we will perform the qualification. So qualification is a process which is very, very important for the instruments which we use in the pharmaceutical manufacturing units and for the equipments. So we are going to discuss the topic of the qualification with an example of UV visible spectrophotometer. So the UV visible spectrophotometer is an instrument that is used to measure the absorbance or the transmissions values of the different solutions over the range of UV which and visible ranges of the electromagnetic spectrum, generally between 200 to 800 nanometers. So the qualification may be defined as act of proving and documenting that and given equipment or process or utility is correctly installed, working properly, and is consistently producing the expected results. So qualification of the UV visible spectrophotometer involves various steps. What are the four steps? One is the design qualification, second one is the installation qualification, and third one is the operation qualification, and fourth one is the performance qualification. So what is meant by design qualification? The type, the make, and the model of the instrument to be purchased must be chosen very carefully depending upon the specific requirements, the type of the sample that will need to be measured. For example, if we need a double beam UV spectrophotometer where the samples simultaneously can be scanned and at the same time the blanking also can be done parallelly. So for that purpose, for that special requirement, we need to purchase double beam UV visible spectrophotometer. So thereby the design is a double beam UV visible spectrophotometer. Okay. So we have to choose based upon our interest, based upon our parameters, and based upon our requirements, based upon our the type of analysis, whether we are doing in a pivotal scale or a pilot scale, and whether we are doing in an preparative scale or analytical scale. So the type and make of the instrument to be purchased must be chosen carefully depending upon the specific requirements of the type of the samples that will need to be measured. So this is about the design qualification. And second one is the installation qualification. Install the instrument in a room that is maintained at the temperature between 15 to 25 degrees centigrade. As because the temperature maintenance is also very important in order to maintain the life of the instrument. So we have to maintain an appropriate temperature that is 15 to 25 degrees centigrade for the UV visible spectrophotometer and relative humidity of 45 to 80%. So the installation site must be away from the dust and corrosive liquids or the gases, and we should not place our equipment directly into the focus of the sunlight, and remove the instrument from its packaging material and place it on a surface that means nothing but it should not vibrate. The surface should not vibrate. So for that purpose, we are going to take an anti-vibrant table and install the computer with an appropriate software near the instrument and check the following parameters. What are the various parameters in the stage of installation? The parameters and the specifications. Appearance. No physical or no defects should be visualized, and all the equipment should be intact. And we have to get the SOP, that is operating manual, that must be present. Parts, all the parts present as per operating manual. Hardwares and softwares that has to be installed, that should be supplied and must be present in this installation qualification. Coming to the operational qualification. First step is that is connecting the instrument to the mains and switch on. Follow the operating manual instructions and check for the following. what we have to check? We have to check for the various parameters such as wavelength accuracy, linearity of absorbance, resolution, wavelength reproducibility, photometric accuracy, sterolite, photometric noise, baseline flatness, and photometric stability. Measure the values obtained for each of the above parameters and check if they met the specified acceptance criteria. Record these values. And coming to the performance qualification, define the performance criteria that are most important to routine operations. And we have to define the acceptance criteria for this performance criteria selected. And we have to determine the values for the chosen criteria and check if they meet the acceptance limits set. So we have to decide on the frequency of regular calibration and performance qualification for routine use of the instrument. Coming to the next topic, that is general principles of analytical method validation. So coming to the definition of the analytical method validation, it is defined as the process of establishing through laboratory studies that the processor's performance characteristics meet the requirements for its intended use. It is not a one-time activity. Methods need to be revalidated on a regular basis to ensure that they are suitable to analyze the materials in use at the point of time. And any changes in equipment or instrumentation or premises may also call for the revalidation or for the revalidation of analytical methods. So steps in the analytical method validation: planning analytical method validation, writing the protocol and getting it approved, executing the approved protocol, analyzing the validation data obtained and reporting the results in an appropriate format of the validation, finalizing the analytical method procedure based on validation results. So coming to the contents of analytical method validation protocol, first one is the objective, second one is the parameters to be evaluated, third one is acceptance criteria for above parameters, experimental details, analytical procedure in draft form, finalizing the analytical method procedure based on evaluation results, procedure to deal with errors and the deviations, methods to be used for the various data analysis. So coming to the various analytical method validation parameters of the characteristics, the parameters that has to be performed for the part of validation programs includes the accuracy, precision, specificity, that is limit of detection, limit of quantification, linearity, robustness, ruggedness, and the range. We all know and we have studied the definition of the accuracy in the first year of the pharmacy itself. The accuracy of an analytical procedure is the closeness of agreement between the values that are accepted either as conventional true values or as an accepted reference values and the values that are found. Coming to the definition of the precision, precision is defined as the degree of closeness of a series of measurements obtained using multiple samples of the same substance under the specified conditions. Precision is defined as the closeness, whether the values are accurate or inaccurate, and how the values in the method validation should be both accurate as well as precise. If the values are accurate, if the values are not precise, and the method validation is said to be failed. If the values are said to be precise but not accurate, in such cases also those values are not going to be taken into consideration. So the values should be both accurate and precise. Coming to the specificity, ICH definition of the specificity is the ability to assess unequivocally and analyze in the presence of other components that are expected to be present. That is, the method that what we have selected should be specific to the components, which is components which are of our interest, not to the other analytes present in the sample, which is of our expertise. Coming to the detection limits. Detection limits or limit of detection is defined as lowest amount of analyte present in a sample that can be detected, but not necessarily quantitated under the stated experimental conditions. So limit of detection is generally expressed in terms of analyte concentration in the sample as per the million or percentage. Detection limit may be established visually or using signal to noise ratios or using data from standard deviations and the slope of a calibration curve. And coming to the quantification limits, limit of quantification. This is about limit of detection and this is about the limit of quantification. So it is defined as the lowest level of an analyte that can be quantitatively measured under the given experimental conditions. Coming to the linearity, linearity is truly based upon the results obtained based upon the concentrations. With more and more concentrations, obviously there should be an directly proportionate results that has to be obtained. So linearity is defined as the ability, that means nothing but a particular range, to obtain the test results of variable data such as the area under curve or absorbance. Nothing but directly proportionality to the concentration of the analyte in the sample. Coming to the range is defined as the capability of an analytical method to remain unaffected by small but deliberate variations in the method parameters. Sorry, the range is defined as an interval between lower and upper concentrations of analyte in the sample for analytical procedure that is demonstrated to possess a suitable level of accuracy, precision, and linearity. So it is nothing but the gap in between the lower concentrations and the upper concentrations. Always we prefer the lower concentrations as the lower concentrations gives an accurate results. And coming to the robustness, it is defined as the capability of an analytical method to remain unaffected by small but deliberate variations in the method parameters. This characteristic indicates how reliable a given analytical method is during normal usage conditions. So finalizing the analytical procedure, the following a successful analytical method validation and the final analytical procedure must be established and documented. The minimum information to be provided in this document includes rationale for the process and procedure and capabilities of the method. If the method is a revised one, advantages of the revision must be described. Complete details of analytical procedure to allow the method to be replicated by anyone reading it. All important instructions and parameters must be maintained here along with formula for the calculation of the results, list of impurities that are permitted and their limits for impurity assays, validate the data, revision history, signatures of authorized personnel and their limits and authorized signatures of the persons from the various departments and QA representatives, reviewers, and everything has to be included. And coming to the need of revalidation for analytical method. Validated methods need to be revalidated in the following situations. When a significant changes has to be done or introduced in the process of synthesizing the drug substances, especially the active pharmaceutical ingredients. When a new impurity is encountered and changing the specificity profile of the method, if the changes are made in excipient composition, which can be introduced with new impurities. And when there is a change in major equipment or change of API supplier, that may change the degradation profile of the API. Coming to the warehousing, good warehousing practices and material management. Coming to the introduction part of the warehousing, warehouse is a suitable place where raw materials or finished products may be stored before their distribution. Proper storage is vital to ensure quality, safety, and efficacy of the pharmaceutical products. All these are should be made for the good manufacturing practices. So the good warehousing practices are also a part of the good manufacturing practices. Good warehousing practice means storing the suppliers so that the products are always available, accessible, and in good condition. Pharmaceutical warehousing is much more than the simple storage of products. We can give a simple definition that simple storage, but it means that actually what it means, it's an operation that preserves the integrity of the drugs as it is when we are receiving and until the usage. According to the current good manufacturing practices, drugs must be stored to prevent contamination and to be positioned to allow for inspection and cleaning of the areas. So each lot of the drug must be identified with a distinctive and traceable. Okay, along with the code and lot number status must be identified. The warehousing official ensures that the storage of each drug is in line which its specific requirements defined by the manufacturer. So what is the importance of good warehousing practices? One is to optimize the resources available for a large scale in a specified manner. And as an integral part of the supply chain to save time and effort in identifying and locating the goods, to maintain a safe, clean, and segregated environment, to control the movement and storage of materials within the stores, to help in easy stock take and stock verification and reconciliation, and help in stock corrections if necessary. Regulatory requirements for pharmaceuticals, to develop a zone concept for product wise segregation, to streamline the process of receival, storage, and distribution. And the key activities involved in the warehousing are warehousing and storage is an act of storage and assorting the finished goods so as to create maximum time utilization at the minimum cost. The key activities concerned with the warehousing area are receiving, identifying, holding, assembling, and processing of the orders to meet the demand. Coming to the elements of good warehousing practices, cost involved in warehousing, it is good to follow the rule of FIFO, first in, first out, or first expiry, first out for the finished goods, which helps to maintain the inventory with the maximum shelf life and stock verification, orderly, timely, and frequent stock verification is the key success for an proper good warehousing practices and which may affect the business positively. And most important one is the safety is which is one of the most foremost important in the warehouse considering the various types of activities and equipment like the forklift, trolley, pallets, drums, shippers, etc. All the employees should wear protective garments commonly called PPEs 22 to protect them from any accidental harm, personal protective garments and the equipments like safety shoe, goggles, hand gloves, and gowns. Everything comes under the safety garments. So premises and the health and hygiene of are very important. The area should be kept clean and away from the objectionable odors, smoke dust, and other contaminants. The warehouse should be well ventilated Opening lead to entry of rodents, circus, birds, and vermin should be closed. Good documentation. SOPs for receiving the raw materials, SOP for appropriate placing, SOP for maintaining the temperature conditions, records, and bin card need to be checked, updated, and religiously followed. Maintain the invoices, delivery notes, and other documents. And coming to the material management, uh, nothing but right quality, right quantity of the supplies at right time, and having good integrity and good inventory control. So there should be a proper planning, organization, and controlling the flow of the materials internally and internal operations, initial purchase, and the service point of the distribution. And there should be an appropriate purchasing of the softwares like LIMS, which are very helpful for an appropriate distribution of the raw materials and what is the stock that we have purchased and what is the stock that is being left and what is the quantity that has been supplied from the various departments. And the material management is a scientific technique which is concerned with the planning, organizing, and control of the flow of material from their initial purchase to the destination. And we will discuss the objectives of the material management and all these things in the next class. Thank you. And if you are having any doubts, you can give your queries in the comment sections, and thereby we'll come back with the clarifications.